This image displays the G protein coupled receptor, the inactive G protein, and the enzyme adenylyl cyclase. The signaling molecule is approaching the coupled receptor. Once it fuses with the receptor changes shape and is activated. In turn, the G protein in the interior moves toward the receptor and binds with the side inside of the membrane. The GDP in the protein is then replaced with a GTP. This activated protein goes to adenylyl cyclase. Here, the protein activates the enzyme that in turn converts ATP into cyclic adenosine monophosphate, also known as CAMP. This CAMP then activates protein kinase A, which goes on to phosphorylate other proteins or set off other triggers in the cell.
The picture here depicts two receptor tyrosine kinase proteins. Signaling molecules known as ligands move toward the receptor proteins. Once they fuse with receptor proteins, these proteins move towards each other. This creates a receptor tyrosine kinase protein dimer. Once the dimer is created, the cell expends 6 ATP to activate the sites of the protein. This triggers the relay proteins, which then become activated. These relay proteins send signals to parts of the cell, including the endoplasmic reticulum. One specific channel on the ER is activated and opened. From here, calcium ions are released into the cell, which cause other cellular responses. In this situation presented, the adenylyl cyclase from the first demonstration is not the correct shape, and therefore is not functional. The G protein cannot bind with adenylyl cyclase, so cellular responses are inhibited. This results in anthrax.